So in Module 5, we will study the muscle tissues, specifically focusing on the anatomy and physiology of the skeletal muscles. So let's begin by examining the characteristics that are common to all muscle tissues, and then we'll make our way back to the skeletal muscles specifically. In general, the muscle tissues compose about half of the body's mass, and all of the muscle tissues are specialized to use the chemical energy within ATP in order to produce movement. When muscle tissues produce movement, they contract or they shorten, which implies that they generate pulling forces. Muscles are unable to produce movement by pushing on the structures that they are attached to. So recall that muscle tissues may be voluntary or they may be involuntary. The term voluntary implies that we have conscious control over those muscles, while the term involuntary implies that we do not have conscious control over those muscles. Muscle tissues, as we discussed in the previous slide, are contractile tissues. So that is, all muscle tissues will contract or shorten in order to produce pulling forces. So that's how they move the structures that they're associated with. Muscles are also extensible, which means they can be stretched to a greater length, particularly when they are relaxed. And they are also elastic, or they have this property of elasticity, which means that they have the ability to recoil back to a resting length after they are stretched. And so in this way, they behave a little bit like a rubber band. So we'll see that there are special proteins within the muscle that give them this elastic property. The muscle's elastic properties do not come from elastic fibers, which we saw in the connective tissues, but it's a different protein entirely that produces the muscle elasticity. So, and finally, muscle tissues are excitable, which means that muscles will respond to a stimulus by changing the voltage that's across its plasma membrane. And this creates an electrical wave called an action potential that will spread along the entire length of the membrane of that muscle cell. The electrical wave, the action potential, is what causes the muscle to ultimately contract. When muscles are not stimulated, they return to a voltage, that is, their membrane returns to a voltage called resting membrane voltage, or another term for it is the resting membrane potential. So in this state, in the resting membrane potential state, they are prepared to contract if the proper stimulus is received. So this diagram summarizes the establishment of the resting membrane potential or the resting membrane voltage. So in this context, the cell is at resting membrane voltage when it is not being stimulated. There are two important substances that contribute to the resting membrane voltage, and those substances are sodium and potassium. When the muscle cell is at rest, sodium is found in higher concentrations on the outside of the cell, which is shown at the top of this diagram, at least compared to the inside, so that is, sodium is found at higher concentrations on the outside of the cell than on the inside of the cell. Potassium is found at higher concentrations on the inside of the cell compared to the outside. So notice that there are two protein channels. One is orange, and that is a potassium leak channel that allows potassium to diffuse or leak out of the cell because the concentration of potassium is high on the inside of the cell. So that means that if it's going to diffuse, that means it's going to diffuse from high to low concentration, and in doing so, it will diffuse out of the high concentration that's inside of the cell. So the other channel is purple, and it is a sodium leak channel that allows sodium to diffuse into the cell. So again, because the sodium concentration is high on the outside of the cell and it's low on the inside of the cell, and so diffusion tells us that sodium will move from the outside of the cell to the inside of the cell, simply following that concentration gradient. We also see another structure in here. It's called the sodium-potassium pump. That's right in the center of the diagram. And the sodium-potassium pump uses ATP to help restore the high concentration of potassium inside the cell and the high concentration of sodium outside the cell. So recall that this is a pump that functions in active transport, and so it must use ATP in order to restore the high concentration gradients of both sodium and potassium. So in reality, there are many more potassium leak channels than there are sodium leak channels in the plasma membrane of these muscle cells, making it easier for very large amounts of potassium to diffuse out of the cell and very little sodium to diffuse in. And since potassium is positively charged, the movement of potassium out of the cell would cause a voltmeter that is placed across the membrane to read negative. So the negative in the voltage reading is really telling us that the potassium, which is again positively charged, is moving out of the cell. So it's simply indicating, the negative is simply indicating the direction of movement. The resting membrane voltage of muscle cells is approximately negative 90 millivolts. This diagram shows negative 70 millivolts because it is a diagram of a neuron in the nervous system. However, the exact same process is occurring in muscle cells.